next speaker is John, as I've said. And John's talk today is developing new microbial chassis and new enzymatic technologies for municipal solid waste valorisation. Over to you, John.
Hello, can you see my screen all right?
Yes, we can hear you. You're sort of not on file slide presentation, but we can see it still, whatever you want to do. There, that's it. Brilliant.
Okay, great. Cool. Thanks for introducing us, Gary. I'm John. So I'm postdoc in Gary's lab at Northumbria University in the building metabolism team, obviously, in the hub. And I'm going to tell a bit what we're working on about developing new chassis organisms and new enzymatic approaches and things for MSW stuff. So obviously, as I've been hearing about loads today, is we're considering in this team group how you can develop buildings that metabolize their own waste. And the approach I've been taking through that is to try and understand some of the existing microbial processes that are already occurring within that municipal solid waste. And then to try and utilize synthetic biology approaches to leverage these to try and make useful things for the house out of some of that waste material. And the way that we started thinking about that was by thinking about the OHM, this futuristic Home of the Future project that Martin spoke about on Tuesday, and I think we're going to hear lots more about tomorrow. And I thought about how might this fit into some sort of futuristic biotech empowered home of the future, and started thinking about how we treat our waste and things at home. We sort of chuck it in the bin when it gets taken away, sort it out into different recycled fractions and things that is picked up by the bin man and stuff. And I thought, well, what if instead you had some sort of futuristic bioreactor inside your house anyway? Because we already sort all of our waste into different fractions and sort of facilitate something like this. So you could maybe have this tiny urban bioreactor inside your kitchen where you can sort your waste materials into lots of different compartments, and in each of those compartments you might have different biotechnological processes happening. You might have engineered microbes in some of them, producing things like materials for you to use, flavorings for your cooking and stuff, and electricity and heat and things to power the home as well. We've also been hearing yesterday, Peter Noende, really cool talk about utilizing some cell-free approaches and stuff, and perhaps that for the concerns that we have for utilizing GMOs and things in a home setting, some of these cell-free approaches might be useful as well in these some of these reaction chambers as well. You might have sort of immobilized enzymes carrying out different reactions, and you might produce things like fine chemicals and stuff like drugs and fuels and flavorings too. So you've been thinking a lot about paper, and you might be thinking, why on earth are you thinking about trying to divert paper to things like fine chemicals and stuff? We can already recycle paper. But paper, actually, you can only recycle it about 4 or 5 times before it sort of loses its usefulness as a recycled product. And actually, there's a huge amount of paper that you might think is recyclable because of the adhesives and dyes and stuff that's on them. They can't be recycled properly. So one of the things that we might be able to use to sort of in order to utilize these this unrecyclable paper properly is engineered microbes and things. So new chassis organisms that are adapted to thrive on this as a substrate anyway. So in order to try and consider that, sort of similar to how John was talking about looking to nature this morning, instead of going out to the beach and things to get these gribble organisms, I went straight out to the bins and got some paper to use for my experiments. So I got from the bins, got some, went to the mixed recycling bin and also the unsorted waste bin, and got lots of different disgusting pieces of paper and things to use in my experiments. And I used these paper samples as both a carbon source and the inoculum in a media that is designed to enrich cellulose degrading organisms. And we cultured those for a week or so. You might see the eagle-eyed amongst you, legendary Newcastle-based baker in one of those bottles. So we cultured them for about a week or so and then plated them out on agar plates that contain cellulose. And then we stained those plates after growing colonies on them with Congo red. And Congo red's a stain that stains cellulose red. So if you can see sort of yellow areas or halos around your colonies and stuff, you have a cellulose degrading organism there. So we screened all of those isolates that we had. We got about 77 different isolates from that, which was a huge amount. Obviously, there might be some redundancy and things in there, but it's pretty cool. So we screened them all for the different properties on how they could grow on chemically defined cellulose, but also on cardboard pulp and things as substrate as well, and some enzymatic tests and things as well, and picked 3 favourites. three favourite strains based on this stream. One of the things that we've done with these strains then after this was to consider each of their preferences for different carbon sources that were relevant to paper and cardboard waste. These are polysaccharides that make up cellulose and hemicellulose. And what this plot shows you is the maximum growth rate, or the smallest doubling time that each of these strains had on each of these different carbon sources. And all that you really take away from this is that they all have sort of distinct preferences. They all, none of them are identical. When we consider the entire profile. And because we could see that, these different preferences like this, start to think about actually, could there be possibilities for synergistic interactions in co-culture, or synergistic interactions on blends of carbon sources in co-culture as well, which is something that we won't talk about today, but we've investigated too. So what happens when we grow these organisms in co-culture? So these plots show us the yield. It's just a sort of approximation of the amount of biomass that we accumulate when we grow these strains in co-culture on each of these different carbon sources. On the far left is what you see in monoculture for the strain US1376. And you can see that when you start growing it in co-culture, the strain US1379, you get, in general terms, a little bit of a boost in yields. But what I'll draw your attention to is the yield, the boost in yield that we see on xylan in particular. And this is something that we've seen reflected in almost all of these co-culture experiments in different pairs. You see a boost on xylan specifically, which has been really interesting. I'll show you that here and here. So this is really cool. So what we did next was to sequence the genomes of these organisms to find out exactly what we had, and maybe try to explain some of these preferences on carbon sources and things we saw, but also to find out exactly what organism they were and how we could begin to start engineering them. And what we found out was that it was possible to transform them with plasmids and things. We've been electroporating them with lots of different plasmids. One of the cool things we've been able to do with them was this optogenetic stuff. So you might have seen Josh's poster yesterday, where he's been working with this optogenetic circuit that responds to blue light. And this one, in the presence of blue light, the expression of a red fluorescent protein is repressed. So when you shine a blue light on them, the colonies stay the same color. But when we grew them in an incubator, sort of side by side, with some plates with tinfoil over them and some without, the ones that had tinfoil in them sort of accumulated all this RFP and got this beautiful pink color. So this is really cool. This is a demonstration that we can do some really cool, state-of-the-art synthetic biology using these strains as well. So what we'd really like to do with them now is start using some of the insight that we have from the complete genome sequences to see sort of what metabolic pathways are existing and how can we sort of upgrade them to make some useful things to use in the home. Okay, so that's a little bit about this new chassis project. I want to tell you a little bit more about some of these enzymatic reaction chambers and what we might do actually using cell-free synthetic biology stuff and engineered pathways and things. The proteins are things that we're interested in using for this are called cellulosomes, and cellulosomes are these really amazing proteinaceous nanomachines that have evolved for the degradation of cellulose, cellulose. And yeah, the reason I have this animation here with your car assembly line with all the robots and things clamping things on is because that's how I like to think about it. Because you have these two different types of protein that make up cellulosomes. You have scaffolding proteins that contain a CBM domain, and this allows them to stick on to Hello,
John. You've just dropped out for a minute there, that's all.
I was talking to myself there. Where did you lose me? I'm sorry.
Yeah, you'd started the slide with the car assembly line, but it dropped out shortly after that.
Okay. Can you see my screen?
Something's happening, but it's not quite there yet.
Hello?
It's not coming through. Oh, it's not on mine.
Can you see my screen?
Yeah, but there's nothing on it. It's just said John Allan.
Dear.
Oh, no, there it is. It's just arrived.
Okay. Hopefully that stays good then.
Yeah. You crack on.
Okay. All right. Okay. I was just talking about these cellulosomes, and they have these two different types of protein called scaffoldins and enzymes that make them up. And the scaffoldin proteins have a CBM domain that allows them to stick really strongly to cellulose and other polysaccharides, and these cohesion domains. And the cohesion domains facilitate a really, really tight interaction to enzymes that have a Dockerin domain on them. So it's a really, really strong interaction. What we see in general is that the different organisms can coexist in similar environments and things, but their cellosomes will be made up of only their own enzymes. So you don't see, so in a lot of cases, you do not see any crosstalk in between the cohesion and Dockerin interactions between different organisms. And this allows you to use synthetic biology to start sort of, programming the length of a pathway. So you could build up each individual enzyme in a pathway in sequence and maybe try and create this synthetic metabolon channeling effect. And cellosomes don't have to be just 5 enzymes big. In nature, they can be up to like the biggest ones that have been described are about 160 enzymes big. And the reason you can get this amazing complexity is because sometimes scaffoldins also have Dockerin domains on them. And these Dockerin domains allow them to sort of dock into another scaffoldin. So a scaffoldin with multiple cohesins can then support multiple bigger scaffoldins. So you can have huge amounts of enzymes building up on there. So this presents some really cool ways for making your own cellulose, as your own synthetic cellosomes. Weirdly though, we've discovered one that is really, really tiny, and in this isolate there's only a single Dockerin domain that is annotatable in the entire genome, and a single cohesion domain, and they're both encoded on the same operon in the genome as well. So this is an indication that this is actually a tiny, tiny little cellulosome that we have here. And the cohesion and Dockerin domains themselves only have about 30% homology to other characterized cohesion and Dockerin domains. So it's indicating they're actually quite unique and quite interesting. So we set about trying to characterize this as a tiny little cellosome system to begin expanding into synthetic cellosomes. We first started studying the enzyme with the Dockerin domain, and we found that this is a GH5 class cellulase. And when we looked at some enzyme kinetics and things, we found out weirdly it has a really, really low affinity for its substrate, which was bizarre. So it has a Km of more than 200 micrograms per mil in these experiments, which was really surprising. And we also did some cool analytic enzymology stuff and found out some interesting structural information about the active site of the enzyme too by using oligosaccharide substrates. And this allowed us to find out that this blue region at the bottom here is a representation of the active site. You see that it has a specificity for 5 glucose residues on a cellulose chain and cuts in between the second and third one. So we want to try and find out why on earth has it got such low affinity, and can you explain that by looking at the scaffoldin protein? And what we found out was that this seems to be the case, that the scaffoldin protein has a role in improving the affinity for the enzyme of its substrate. So you see it, and specifically on insoluble substrates as well. So on the left-hand side here, this graph is the specific activity of the enzyme in the presence and absence of the scaffoldin on soluble cellulose called carboxymethyl cellulose. But next to that, we have the specific activity on insoluble microcrystalline cellulose. And you see this, the specific activity almost doubles in this case. So that's really cool. That was a really nice result. But we've also been able to demonstrate that the scaffoldin protein can interact with this insoluble cellulose as well. So when you see here on these gels, when you increase the concentration of microcrystalline cellulose, you get more and more of it pulled out. And it doesn't seem to matter how much scaffoldin you put in, you always get, it always sticks to the cellulose if you keep the cellulose concentration the same. This all sort of builds towards a model of this actually being a functional tiny little cellulosome, where the scaffoldin just sticks the whole complex onto the polysaccharide substrate, while the enzyme sort of munches it off into lots of little bits. So I'll leave that there for now. The ongoing work from this is studying sort of using those chassis to start making some interesting products and things, and putting some cool enzymes in them. And with the Cellulose project, what we're working on at the moment is sort of studying the interaction between the two proteins and trying to sort of find out actually what's the physiological relevance for this organism to have such a tiny one, and then begin making synthetic cellosomes with it. So yeah, that's all from me. Thank you very much. I'd love to hear any questions. Oh no, I've got some thank yous from everyone in the lab and things, and to Greg, who's posting, might see on Tuesday, and Andy and Darren, who did lots of the genome sequencing for us. and these guys as well, who are great help in the lab. Yeah, thanks for it. Thank you.
Brilliant. Thank you very much, John. Unfortunately, because we lost you for a minute or so, we probably lost time for questions. But again,
that's okay.
If people could stick questions into the chat, I'm sure John will respond, or email him, or he's normally found by the bins somewhere, really. So thanks very much.
The bins or the pub. Yeah. Okay, thank you.
Thank you.